Welcome to this Molecular Plant Pathology podcast. I am your host, Anadjasa M. Ocho, together with
I am Charles P. Cabrera,
a fourth-year plant pathology students of University of Southeastern Philippines. This podcast will talk about molecular aspects in the interaction of Ustilago maydis with its host plant causing corn smut disease. Now let's talk a little about corn smut. So corn smut is caused by the fungus Ustilago maydis, and it occurs wherever corn is grown. Mainly, it damages plants and had significant impact on crop yield, particularly because of the formation of galls on the above-ground parts including ears, tassels, stalks, and leaves. What's sadder is that maize is one of the world's most important cereal crops. Thus, this disease caused significant losses in production quality, putting global food security at risk. So, Charles, we're talking here about Ustilago maydis. Tell us more about its molecular genetics.
Yes, numerous investigations in the last decade have identified Ustilago maydis cellular and metabolic control of the maize host throughout the infection and tumorigenesis process. From comparison of the process in floral and vegetative organs, it is clear that Ustilago maydis activates plant DNA synthesis and cell cycle in differentiated leaf cells and induces divisions in specific cell layers and cell types to form tumors.
Indeed, it's interesting to consider that the activation of DNA synthesis and the cell cycle promoted tumor formations in cells. Moved on to the effectors of Ustilago maydis. So, what are the secreted effectors of this pathogen?
Sequencing of the Ustilago maydis genome and transcriptional profiling of different infection stages paved the path for the discovery of effector genes that govern interaction with the host plant. Effectors are broadly defined as microbe-derived secreted molecules that shape interaction with the host. A recent study revealed that effector genes are differentially expressed in different infected maize organs, and that some effector mutants affect tumor formation only in specific organs. This mirrors the finding that different maize organs express distinct sets of proteins and suggests that the ability of the Ustilago maydis to induce tumors in different maize organs relies on its ability to reprogram different plant developmental states to suit its own needs.
So, how about those effectors that is present in the Ustilago maydis hyphae and its host?
Effectors acting in the biotrophic interface between the Ustilago maydis hyphae and host. During the early phase of maize infection, Ustilago maydis is recognized by the plant via conserved molecular patterns. This leads to salicylate-dependent defense responses, a typical response of plant to biothropes. These plant defense responses are considered to be overcome or suppressed with the help of the set of core effector molecules that are upregulated during penetration. One of these core effectors is PEP1. PEP1 mutants are able to form normal appressoria but are arrested during penetration and induce strong plant defense responses that include the massive transcriptional upregulation of the maize secreted peroxidase POX12 and an accumulation of H2O2 at attempted sites of penetration.
That's interesting. I have also read some articles talking about the effector of the host of Ustilago maydis acting inside plant cells, and it talks about plant pathogen systems where respective analyses have been done. The repertoire of effectors consists of those functionings in the apoplast and those taken up by plant cells. In Ustilago maydis, a secreted charismate mutase or the CMU1 serves as the first example of an effector that is translocated into the host cell. The CMU1 gene of Ustilago maydis is among the most strongly induced genes during plant colonization, and the CMU1 protein is the most abundant fungal protein detected in the apoplast.
Based on that articles that you've read about the effectors of the host of Ustilago maydis acting on the plant cell, there is this interface between Ustilago maydis hyphae and host as the site for obtaining nutrients. For acquisition of nutrients, biotrophic fungi have to divert the metabolism of the host to provide them with nutrients via the extracellular biotrophic interface. This mechanistically not trivial, as the extracellular release of monosaccharides has been shown to trigger plant defense responses. With more genome sequences of related SMUT fungi with highly synthetic genomes becoming available, the identification of conventionally secreted effectors has become an easier task, as many of these genes are only poorly conserved.
So, is it possible that comparative genomics also allow for identification of effector sets that might have a role in determining host specificity?
Yes, because comparative genomics is a valuable tool for defining conserved effector domains for functional assays.
And I have heard that Ustilago maydis have already been identified and characterized based on their transmembrane proteins for virulence. Can you tell us more about it?
Based on identification and characterization of two transmembrane proteins required for virulence of Ustilago maydis, they found out that two of the six proteins named VMP1 and VMP2 are essential for the full virulence of Ustilago maydis. The deletion of the corresponding genes leads to substantial attenuation in the virulence of Ustilago maydis. The biochemical analysis clearly shows that VMP1 and VMP2 are membrane-associated proteins potentially localizing to the Ustilago maydis plasma membrane. Mass spectrometry and light scattering suggest that VMP1 mainly occurs as a monomer, while VMP2 is a dimeric. Notably, the large and partially unstructured C-terminal domain of VMP2 is crucial for the virulence while not contributing to dimerization.
Now, this is my last query, and this probably the most important to ask about the Ustilago maydis causing corn smut disease, which is its proper management approach.
Some studies states that no corn varieties or hybrids completely resistant to smut are known, but several corn hybrids show moderate resistance to the fungus. New pathogen races appear constantly, however, therefore partial resistance is the major type of resistance selected for the breeding programs. Sanitation measures such as removal of smut galls before they break open and crop rotation help where corn is grown in small, rather isolated plots, but is impractical and impossible in large corn growing areas. But based on my statement earlier, dealing with the molecular aspects of the interaction of the host and the pathogen, it serves as an opportunity for the development of new and more concrete management technique.
Undeniably, hoping that the interaction between the host and the pathogen provides a chance for new and more concrete management techniques to be developed, as Ustilago maydis really is a very important disease in plant. And because of this, knowing their molecular aspects, most especially the different effectors and the character of Ustilago maydis, surely they have an idea to combat this disease. Thank you, Charles, for imparting your knowledge and participating in this podcast.
I am so grateful to share this knowledge to listeners, especially those who are into plant disease management, in the hopes to combat the existing problems with the goal of eradicating the disease, which is one way in achieving food security. I've enjoyed this podcast and I hope we had another chance to talk and discuss other topic relating to molecular plant pathology. So, thank you also for having me here. Once again, this is Ian Charles Cabrera.
Yes, we're also looking forward for another informative talk with you, Charles. Thank you everyone for listening to this podcast. Stay tuned for our next episodes of Molecular Plant Pathology Podcast. This is Anajasa M. Ocho, wishing you all a happy holiday. Thank you!